Morvezen, working for SYSAAF. I'm a genomic breeding program manager, among other stuff I do. And today I'm going to talk about the application of genomic selection in breeding programs with SYSAAF. So that's a direct application of results from research projects, including AQUAIMPACT and AQUA-FAANG. So just to remind you what is, remind you, or maybe you will learn, what is SYSAAF, where I'm working. It's a unique collective and professional organization. We are an association of 32 breeding companies, mainly French, but not only, including 18 aquaculture companies. We're a team of 24 geneticists, data scientists, and engineers. We currently have 2 PhD candidates. We're basically a service provider in selective breeding. So if you're a hatchery, if you're a breeding company, we can help you implement genetic tools, breeding programs, and help you optimize them. We work with genetic or genomic evaluation of 119 lines of 29 different species of poultry, aquatic, and insect species. The insects being the earlier one. And we currently have more than 7 million individuals in our databases just for aquaculture. We collect more than 2,500,000 new data points per year. And we've started working 70 years ago. Well, obviously not me, but the SYSAAF is has been existing for 70 years, 71 actually. So I want to give you an overview of the implementation of genomics and the impact of genomics on the breeding program for the SYSAAF. So it's a long story of different research projects. The first one was Fish Boost that started in 2014, was like the founding project for us. It was already a big European consortium project, and based on that project and on the first results that were published, we collectively made the decision to invest in genomic selection in 2015. And then I'm not going to detail all the different national and European projects that we took part of, or the members took part of. But it was a long R&D era. And before 2020, we developed tools. Well, we're still developing tools. We implement them in the breeding program for the members, and we have the first genomic evaluation. So the first actual animals being born selected by genomics, starting in 2018. So it was quite a revolution, quite a quick one. The genomic tools arrived around 2010, and less than 10 years after the first animals were born using those tools. And now, since 2020, I like to say, it's very just my opinion, we're more in the efficiency and cost effectiveness era. So now we want to do more precision breeding, such as was presented by Sigborn for AQUA-FAANG. We also want some more cost-effective tools. So Adrienne mentioned multi-species array. We also want to do smaller density panels. Less SNPs is less expensive, and we want to make them as efficient as higher densities arrays. And so that's what we're focusing on, improving the efficiency and the cost effectiveness of the genomic breeding program that we do. So what genomics has changed? So what the different research programs, including AQUAIMPACT and AQUA-FAANG, have changed for the SYSAAF. So from SYSAAF side, it has led to a strengthening of the team. We hired 3 geneticists, myself included, and we co-supervised 4 PhD theses. The PhD students worked or are working on the implementation of genomic selection in commercial breeding program. So it's a need that we have to have some research to implement them directly in the company breeding programs. It has, of course, we have developed and adapt data treatment pipelines and softwares. We have had need for reinforcement of informatic capabilities. We now have partnership with large computer clusters to do the actual model, to do the computation, because it's very intensive calculations. We also have a partnership with genotyping and genomic sequencing companies and labs, such as, well, you know them, Thermo Fisher, Illumina, and the genotyping labs. So that was from our side, and from our members' side, so the breeding companies. They have switched 10 genetic lines to genomic selection for 5 species, the 5 I've mentioned, so rainbow trout, sea bream, sea bass, oyster, and shrimp. It was actually pretty easy for them to implement the genomic selection because they were already doing genotyping for parentage, like NT presented. So they were already doing genotyping for pedigree reconstruction. So they basically, some of them just had to switch genotyping technology, send their samples in another lab, pay a bit more, and get some genomic data. And the collective aspect of SYSAAF made that the different members could share the know-how, the genomic tools, and it was very easier to transfer between species the development knowledge, like what work, what don't work, etc. That's one of the strengths of SYSAAF and one of the strengths of participating in big European projects, because, well, you have a lot of partners that have all their experience that you can build on. And nowadays, we genotype nearly 30,000 samples per year. Well, not ourselves, but the members. And we have sequenced more than 700 whole genomes from broodstock from the breeding companies for different research purposes. So it was quite a quick and intense switch when the genomic revolution happened. And now a quick focus on 2 results of the project. It's just an example of what we get from the European projects. Of course, I didn't pick them at random. The first one is from AQUAIMPACT. In AQUAIMPACT, we tested a new, well, not new, but a method for cost effectiveness genotyping. That's the imputation. So imputation is basically genotyping a 1000 SNP panel, so it's less costly, and doing imputation to make it as efficient as full 55,000 SNP array. You can see here for different, so this is for sea bass, for different traits. We get the accuracy in gray is the one that if we only have the pedigree, in orange is with the SNP chip, a full SNP chip, and in blue is with the imputation. So with the imputation, we are nearly as good as with the SNP chip, but it's a lot less costly. So we can make good genomic selection for a lot less money. So you can either have more efficiency or more genotypes. And another example is from, actually it's from several projects. We identified the QTL for sea bass for VNN resistance in the GenC French project. It was then refined, and we found a major gene that is a very good candidate to be a causative gene, and we are looking at the causative mutation right now. It's called EFI6. You can see the paper if you want some details. It does give very good resistance, like you have here for the genotype at this QTL, the percentage of survival. So if you have both resistance allele for this population, for example, you survive at 80%, and if you have both sensitive, you survive at 40%. So it's very substantial QTLs, and we were very happy to see that in AQUA-FAANG, another team, other partners on another population, found the same QTL and arrived at nearly the same gene in AQUA-FAANG. So the work in AQUA-FAANG will help us refine this QTL and do more precision breeding with lower density SNP chip. So just 2 examples. I could probably go on for more than an hour showing you example of direct implementation of results from European projects. And that was all for me. If you have questions.